give you a general overview of what is being done in Heidelberg in the field of virotherapy, and the purpose is that this may trigger some interactions with the Grand Est. We have a long tradition of cooperation with the Conservatoire du Grand Est, and we would be very happy if we could start new cooperations in this area. So I'll just briefly introduce the people from Heidelberg who are present today. So we are working with two oncolytic viruses in Heidelberg, the parvoviruses and measles virus. And the parvovirus community is represented by Asia Angelova, who is sitting there on the side, Antonio Marchini, sorry, and myself, and Christine Engeland is, sorry, I lost my voice, and from time to time I will need maybe a glass of water to recover. And Christine Engeland will, thank you. And Christine Engeland will represent the work, will present the work done on measles virus. So I would like to, for those who are not very familiar with the field of oncolytic virotherapy, to give a short introduction for 10 minutes or so, and then there will be three presentations, one on the natural wild-type parvoviruses used as oncolytic agents, a second one on modified parvoviruses, strategies to improve them, and then a third presentation on the work done with the measles virus, with a focus on the immunological aspects of immunovirotherapy. So let's start with a general introduction. If it's not necessary, stop me. If it's trivial, we can gain time, and you can certainly stop me. So we are going to focus the presentation on the uses of viruses as cancer therapeutics, and the key word of oncolytic virotherapy is oncoselectivity. These viruses must have a selectivity for tumor cells. Otherwise, they would be of no therapeutic value, and this is somehow, I will not go into any detail, but this is illustrated by this slide. As you know, tumor cells differ from the normal cells by a number of properties. They proliferate in an uncontrolled fashion. They have pathways allowing them to resist death under stress conditions. They have altered innate immune responses, so they can not they are altered in their capacity to fight against the virus. So there are a number of properties distinguishing tumor cells from normal cells, and some viruses take advantage of that to replicate preferentially in the tumor cells. So a number of viruses, I would say a dozen of viruses has been identified which show oncoselectivity. And some of these viruses are naturally oncoselective. It is the case of the parvoviruses. Others have to be selected, adapted. So for instance, the vaccinia or the measles vaccines are somehow adapted in that direction. Others have to be engineered to acquire the oncoselectivity. So some viruses are naturally oncoselective. Others have to be modified to acquire this oncoselectivity. But we are in the first two presentations going to speak about the parvoviruses, and these are naturally oncoselective. So some viruses are endowed with an oncoselectivity, and this leads them to kill preferentially the tumor cells in which replicate compared with the normal cells. And this is the paradigm of oncolytic virotherapy. The virus either it doesn't penetrate in a normal cell, or if it has penetrated it, it doesn't kill it because it doesn't replicate, and on the contrary, in the cancer cells it will replicate, kill the cell, therefore leading to the release of progeny particles which can infect neighboring cells, and so there can be waves of successive waves of infection of tumor cells. And a dozen of viruses have this property, but this is not the only key to the therapeutic or the oncosuppressive effect of these viruses. The oncolysis is important, it is necessary, but usually it is not sufficient to lead to the suppression of tumor. Usually in the in vivo situation in humans, the propagation of these viruses is not very, very efficient, and this process by itself does not account for the suppression of the tumor. Actually, the suppression of the tumor relies on an immune response which which is initiated by this oncolytic event and then takes over and continues the job. And this is a process that oncolytic viruses also actively induce. They induce an immunogenic type of cell death. So they actively favor the induction of an anti-tumor immune response. Of course, by lysing, so this is a tumor cell infected with virus, by lysing these cells, the virus will favor the release of tumor-associated antigens, but it does more than that. It induces an immunogenic type of cell death with the release of the so-called DAMs and PAMs, the damage and the pathogen-associated molecular patterns, which will induce the maturation of antigen-presenting cells, the recruitment and activation of various immune effector cells. So eventually, the death of the induced by the virus, the death of the tumor cells induced by the virus, results in a local inflammatory reaction, which is favorable to the recruitment and activation of immune cells. So a second and usually even more important key to oncosuppression by oncolytic viruses is this activation of an anti-tumor immune response. But of course, it depends on the initial oncolytic effects. And there are models where oncolysis is very efficient and you don't need any of this, but in most models, oncolysis is limited, and therefore the success of the suppression of the tumors depends on this immunological factor. Now, so in a sense, virotherapy is a form of immunotherapy. It is considered nowadays in all immunotherapeutic meetings, one speaks about virotherapy because somehow virotherapy involves an immunotherapy component. And oncolytic viruses are very popular presently in the immunotherapy field, just by themselves or in combination treatments. And this is one example of the combination which is being explored and which proved to be efficient in a number of cases. You know that presently a very hot topic in cancer immunotherapy concerns the checkpoint inhibitors, that is, checkpoint factors which downregulate the immune response and contribute to the escape of the tumor from the anti-tumor immune response. And these they are very famous, and you certainly know PD-1, CTLA-4, they are known checkpoints inhibitors, checkpoints factors, and therefore to prevent this downregulation, a number of strategies have been developed, antibodies have been developed against these, and these antibodies indeed boost the immune response and improve the anti-tumor effect in certain cases, but not in many cases. In a number of cases, these antibodies do not help because the tumor cells, the immune cells, remain at the periphery of the tumor, and if you activate them, it doesn't help much because they don't reach their target. And the idea is to combine these antibodies with oncolytic viruses, which will attract and activate already to some extent the immune cells within the tumor, and in that case the antibodies will be efficient because the immune cells will be located at the right place to exert their effect. So the oncolytic viruses are considered either by themselves, but especially in the framework of combination treatments presently in the field of cancer immunotherapy. Another approach is the combination of oncolytic viruses with adoptive cell transfer, for instance, and I think that Christine will illustrate some of these aspects later on in her talk. Good. So presently, as I said, a dozen of different viruses have been shown to have these oncolytic and immunostimulating properties, and they belong to different viral families, which are listed here, DNA, RNA viruses. And today we are going to speak about the parvoviruses. We are looking at both ends. We speak about parvoviruses, which are DNA viruses, and measles virus, which is an RNA virus. Now, these viruses are in clinical trials, so the preclinical data were promising enough to lead to the testing of these viruses at the clinical level, and there are different stages of clinical evaluation. Some are in phase 1, others phase 2, and some reach already the phase 3, and 2 of them have been approved, an adenovirus in China, and recently a herpes virus by the FDA in the States, and so these are now medicinal drugs which can be purchased in the pharmacy. So the field is not simply wishful thinking. The data are convincing enough, leading to the approval of some of them as pharmaceutical drugs. this is an example of the data which led to the approval of this herpes, oncolytic herpes virus. Here we see the case of a metastatic melanoma patient, and the virus was inoculated in one of the nodules, and this led to the regression of that nodule, but also to a bystander effect. Distant nodules have to also metastasis in the lungs at different location in the treated individual disappeared, as shown also here. I think one of these was neglected, and you see they all disappear, and this the metastases are also regressing. So there is a local effect, but also a bystander effect, and on that basis, several of these products reached already the phase 3 up to the approval stage. Now, because I want to be short, so this summarizes roughly the state of the art. More than 1,000 patients have been tested, so an increasing number of trials are being conducted with oncolytic viruses, and these trials show the safety of the viruses compared with conventional treatment. These viruses are extraordinarily safe. The proof of principle was shown. One has demonstrated that there is indeed a lytic infection of tumors, and first evidence of tumor suppression in terms of stabilization, regression of tumors, both local and systemic, have been demonstrated. So the proof of concept has been provided not only in preclinical models, but also in patients. But there is clearly a need for improving potency, and that's why we are here today, and that's why this topic raises so much of interest. The data are very, very promising, but this is an area which is still emerging. It is an area of very intensive research and development. We just came back from an oncolytic virus meeting. Now dozens of new companies have been established with a focus on oncolytic viruses. Big companies like Amgen and others have a priority which is associated with oncolytic viruses. So the data are very promising, but it's not the final word. There is a need for improving therapeutic potency, and that's why there is presently a search for new viruses, analysis of modification of viruses or combination of viruses with other treatments. So there are a lot of avenues are being explored to improve the efficacy of the on the virotherapeutic approach. And we would like today to illustrate what is done in this area in Heidelberg. And in Heidelberg, the work on oncolytic viruses is performed at 2 levels in a research institution, the DKFZ, so the German Cancer Research Center, and at the clinical institution, University Hospital, and the associated NCT, National Center for Tumor Diseases, which is a little bit like a centre anticancéreuse. And so these are the two, and we of course cooperate at the DKFZ at the level of the research and development. We study parvoviruses, and at the NCT, Christine Engeland is working on measles virus. The clinical applications are performed here in the clinical area of Heidelberg. In the case of parvoviruses, the target tumors for the clinical applications are glioblastoma, pancreatic cancer, CRC, and neuroblastoma, and for measles virus, different solid tumors. And in addition, we have already alliance with other institutions, and I mentioned this since one of the purpose of the meeting is a little bit political, how to create alliances. And I have to mention that we will cooperate very soon with the Luxembourg Institute of Health, where Antonio Marchini will, thank you, I think it would be necessary, where Antonio Marchini will work part-time starting the next year. And in Luxembourg, Antonio will further work with parvoviruses, at the beginning at the level of research and development, but there are trials in the pipeline. And another alliance with the hospital in Darmstadt, where the neurosurgeon, Carsten Geletnicki, who was involved in the trial in Heidelberg, moved recently and is further performing clinical work using parvoviruses against glioblastoma. So this is the present scenario, and the purpose of today's meeting is maybe to add another column with the canceropole and or teams associated with the canceropole, and to extend these alliances. So there will be, I'm sorry to go fast, but I think we are short of time. So there will be 3 presentations to illustrate what we do. The first presentation will be on and will be focused on the clinical. I will not speak too much about basic research, but on the clinical applications of natural parvoviruses. And this is the work performed in my lab. I will be here speaking as the ambassador of AstraZeneca. We did a lot in this area, and I'm going to illustrate the results of the accompanying research, the research which accompanies the trial, and she's the main actor of that. And then there will be a second presentation of Antonio, which will deal still with parvoviruses, but how to improve their efficacy through modification or combination, and then a third presentation by Christine on the oncolytic measles virus. So, after this short introduction, I would like to start and speak about the natural parvoviruses. So the story I'm going to tell you now involves a virus which is not modified. It is a natural wild-type virus. Shall I start, or you have already, if you have questions, please interrupt me. Shall we proceed? No? Don't hesitate to stop me. Yeah? Okay, so I would like to illustrate clinical applications of oncolytic H1 parvovirus. We did a lot of preclinical work, but I'm not going to speak about that. I would rather want to illustrate the clinical applications, hoping to raise some interest from the clinicians also. The virus we are dealing with is called H1PV, H1 parvovirus, and it's a proto-parvovirus, it's a rodent proto-parvovirus. Its normal host is the rat. It is the smallest of the oncolytic viruses presently in use. So the diameter is about 20 nanometers, and the size of the genome is 5,000 nucleotides. So it's very, very small. And the viral cycle is depicted here. Most of it takes place in the nucleus of the cell, where the replication, expression of the genome, and eventually the assembly, packaging of the particle take place. And I just circled here steps of this life cycle to show that these were shown to be stimulated as a result of the malignant transformation of host cells, the abnormal proliferation of the cells, the activation of metabolic pathways allowing tumor cells to resist stress. These changes are used by the parvoviruses and lead to a stimulation of their replication. So this cycle, as shown here, takes place in human cells, but not in a normal human cell, but in malignantly, neoplastically transformed cells. But I will not show you the evidence for that, but there is a selectivity of the virus. And as you will see later on, this virus is totally safe. It's not a virus which will attack normal human cells and cause any problems in humans. So that's enough for the virus, and we'd like now to illustrate 3 clinical applications. The one which is the most advanced, because it was the object of a first clinical trial which has now been completed, concerns glioblastoma, and I would like to illustrate the result of a first phase 1/2A study of this H1PV in patients with recurrent glioblastoma. And as I said, this trial is called Parvorix, because the sponsor of the trial is called Oryx, and Parvo because the virus is H1 parvovirus, and it's a cooperation between our department and the Department of Neurosurgery at University of Heidelberg, where the main actor is Carsten Geletnicki. So why glioblastoma? We did a lot of preclinical work, and I will not show it in detail. I just summarizes the main conclusions that we obtained at the preclinical level. First, we showed that human glioma cells can be killed very efficiently with H1PV, and this killing process turned out to be very interesting because it involves the induction and cytosolic relocation of lysosomal proteases, catepsins. So there is an induction of catepsins, and instead of remaining confined to the lysosomes, these catepsins get relocated in the cytosol. And this accumulation of proteases in the cytosol leads to the death of the cells. And so this is a bit of an unconventional death pathway, and it allows the H1PV to kill glioblastoma cells which became resistant to conventional death inducers, for instance, trail or cisplatin inducers of apoptosis. As you know, one of the problems in cancer therapy is the acquired resistance of the tumor cells to these genotoxic agents, and the parvo circumvents this resistance by mobilizing this type of pathway. But it's not the only killing pathway that the virus can trigger. It can also induce apoptosis. It can also induce other types of death, and this is one of the strengths of the virus. If the cell resists one pathway, the virus can still kill it through an alternative pathway. So human glioma cells in culture, including short-term human glioma cells, are quite sensitive to the virus. Then we had, of course, animal models, and we had an orthotopic rat glioma model, and in this orthotopic rat glioma model, the virus proves to be very efficient, leading to a full remission of the tumor after both intratumoral, local, but also intravenous or intranasal administration of the virus. And so in this model, which is a rat model, the rat being the natural host of the virus is extremely efficient in causing the full remission of the tumor in absence of any side effects. So this is a very clean process. In normal tissues, nothing is happening. Now, as I said previously, the immune component is important for the induction of tumor suppression, and this is also the case in this model. For instance, if you deplete the CTLs, you see that the virus puts a brake on the growth of the tumor, because this tumor would explode within this period of time. So there is a brake on the tumor, on the growth of the tumor, but there is no full remission. So the full remission involves the virus, but also the immune system. And under immunocompetent condition, the remission leads to results in a therapeutic vaccination. So the animals get vaccinated against the tumor. There is induction of an anti-tumor immune response. So human glioma cells, glioblastoma cells are sensitive to the virus, and in model system, the virus can be very efficient. In xenograft model using human glioblastoma cells, the virus is also efficient. So there was a series of preclinical data supporting the efficacy of the virus against glioblastoma, and this led to the launching a long time ago. These stories take a lot of time. It's 4 years ago, of a phase 1/2A analysis trial in glioblastoma patients, and I would like to illustrate some of the results of this trial at the research level, and I'll give a few conclusions regarding the clinical outcome of the treatment. So it is a phase 1/2A, so it's an early phase. There are no controls. It is a non-controlled study. There are the patients concerned are suffering from a recurrent glioblastoma. So the glioblastoma was already operated once. It recurred, and the lesion was progressing in spite of conventional therapies. And the patients were subdivided in 2 arms. In one arm, the virus was injected through an operation, so it was injected intratumorally, and in the second arm, it was administered through repeated intravenous infusions. And then in both arms, after 9 days, the tumor was resected, and the virus was again administered around the cavity. So intracerebrally in the cavity, with the hope to reach the remnants of the tumor that the surgeon could not remove. So this is probably not an optimal scheme to reach anti-tumor efficacy, because the tumor is removed rather shortly after the virus treatment. And if you need indeed a tumor cell factory to produce a virus to induce the immune response, probably you need more time. But this scheme was imposed by the regulatory agencies, and it has also the advantage of allowing us to have infected tumor material for analysis. And I would like now to show the results of these is analysis of the research which accompanies the trial. So now I'm going to show data from the resected tumors which were resected 9 days after the virus treatment. And there are several take-home messages from this trial, and I just would like to illustrate one of them here, which is that the virus is able to cross the blood-brain barrier in both directions. So if the virus is inoculated in the tumor, so local injection, after a while the virus is transferred to the bloodstream and there is a transient viremia. So it can leave the tumor and reach the bloodstream. But conversely, if the virus is administered intravenously, you have here, I told you that there were 5 infusions, so you have 5 waves of viremia, and this is followed by the penetration of the virus into the tumor from blood vessels supplying the tumor, and this is evidenced here by this red labeling. This is a fish labeling, so an in situ hybridization, virus-specific in situ hybridization, revealing viral nucleic acid imprints in the cell, and you can see that indeed the virus enters and infects the tumor cells after IV administration. And this is, of course, an asset. It's a big discussion in the meeting. What is better, intratumoral or intravenous? Probably the combination is the optimal scenario. But of course, the intravenous treatment is much less invasive and also gives hope of reaching disseminated tumors. So it's certainly an asset of the virus to be able to enter the tumor from the bloodstream and propagate in the tumor. Now, the vasculature of the gliomas is, of course, altered, but in the rat model, we could show that this transfer from the bloodstream to the cerebral tissue takes place irrespective of any tumor. So the virus per se has an ability to cross the blood-brain barrier, and this was confirmed in the human situation. Now, what's happening once the virus is in the tumor? It spreads. One finds the virus all over the tumor. So it's a very small virus, so it doesn't stick where you inject it. It goes everywhere in the tumor. And what does it do there? It replicates, at least to some extent. And these are, again, analysis of tumors resected from virus-treated patients. And the upper row shows fish evidence of viral RNAs. So again, using the right probes, you can reveal the accumulation of viral RNAs, and you can see that in the tumor there is viral transcription, and the cells which support this transcription are cells having all the distinguishing features of the glioma cells. Gliomas, like many tumors, are very heterogeneous. A major component of gliomas is non-tumor material. It consists of microglial cells, macrophages, but if you label for these cells in green here, you see that the cells supporting the replication of the virus are not the macrophages, they are actually the tumor cells. Now, so there is transcription and also expression of the viral proteins, and here this is a protein we are much interested in and did a lot of work in and still continue to work on in the lab. It is a viral protein called NS1, which is produced in the infected tumors in the patients, and this protein is important because it is an oncotoxic protein. We showed that it has by itself, it can cause the death of tumor cells in an oncogene-dependent fashion. So it has oncotoxic properties, therefore it's important to see that it is produced in the patients, and interestingly, the cells, these are tumor cells, so the tumor cells expressing this protein cluster in the vicinity of necrotic areas, so one may speculate that this expression of the viral toxic protein plays a role in the formation of some of these necrotic foci, but all that is, of course, these are not experiments that you can control. All what I say has to be interpreted with caution. A hallmark of gliomas is necrosis, and therefore you cannot demonstrate here that the viral proteins are responsible for the necrosis. But it's striking to say that these oncotoxic proteins accumulate in close vicinity to necrotic areas. Now, another and probably the most important feature observation made in this trial is that the replication of the virus, the expression of the virus, the production of the viral oncotoxic protein is associated with changes in the tumor microenvironment. So we are not going to speak about the tumor cells now, but cells from the microenvironment. And one thing is that there is around at the places where virus actively replicates and gets expressed, there is a local inflammation, there is activation of macrophages. And this is, for instance, an area of NS1 expressing tumor cells, so an area where tumor cells are actively infected with the virus, and these cells were here stained for the catepsins, which I told you is involved in the killing of the tumor cells. And indeed, one sees, this is the red staining, one sees that tumor cells express catepsins, and if what is observed in vitro applies here, these cells are doomed to die. But the other message is that other cells are also overexpressing catepsins, and these cells are double labeled in red, so they overproduce, like the tumor cells, they overproduce the catepsins, but also in green for a macrophage marker. So these are macrophages, and it shows that the macrophages around the infected tumor cells get activated and produce, in particular, high levels of catepsins. And it was reported in literature that indeed activated macrophages and microglia produce different factors, including catepsins, so lysosomal proteases, which are released from the cell and are toxic for the neighboring tumor cells. So there is a local inflammation, which is probably an antiglioma microenvironment. And the second and important thing is that this microenvironment also appears to be immunogenic. There is a massive infiltration of the infected tumors with leukocytes. So you see here in red the tumor cells, which are fish labeled for viral transcripts. So these are infected tumor cells which express the virus, and in green you have this green means just red blood cells, so these are vessels, and here you see around these vessels in green the leukocytes which cluster around the vessels, penetrate the tumor, and then get scattered around the whole tumor. So there is a massive infiltration of immune cells, and these leukocytes appear to be lymphocytes, both CD4 and CD8 lymphocytes, and very interestingly, they are activated. They express interferon gamma, IL-2, and also the CTL markers, granzyme B, perforin B. So you have a mass of activated T cells in the gliomas, and this is not something that you see normally. When we spoke with the pathologists, they first told us, oh, your interferon gamma labeling must be an artifact, one never sees that, but this always was confirmed, including by them. So there is a massive modification of the microenvironment in the direction of an immunogenic microenvironment. Now, of course, the question is what is the specificity of these immune cells? We couldn't analyze it in situ in the tumor, but we analyze it in the peripheral blood by using the technique of interferon gamma ELISpot assays, which allows to show the induction of CTLs responsive to defined antigenic determinants. And if you look here at the colored columns, you see that there is with time an induction of an antiviral immune response, which is directed against both NS1 and VP proteins. So in the tumor, there is expression of the NS1 protein, and there is also a T cell reaction against this protein and also against the VV protein with time, and this is persisting for a certain amount of time. So there is induction of an antiviral cellular immune response. So of course, is this good or bad? It is a double-edged sword. By having such a response, you will somehow